Well, thank you very much for the opportunity to present at this webinar. My name's Alastair King. I'm the head of biology at Eurofins Discovery. And I'm going to speak to you today on how we as an industry are addressing the challenges of oncology drug discovery, and really at all stages of drug discovery. So looking both at the targeted side of things, the challenges faced there, as well as the challenges with other aspects of drug discovery. This is a little bit of a background about me. I have a background in big pharma drug discovery. I came to Eurofins about 10 years ago, having led drug discovery teams with a focus in specifically the oncology and the inflammation areas. I have had the privilege of leading teams, and that has resulted in several drugs that have been approved from market. So what we'll look at today and what we'll learn today will address some of the key challenges in oncology drug discovery, what they are, what are those challenges that make oncology drug discovery difficult, and how are we collectively addressing them? How are we resolving those challenges? We're going to take a quick look at some of the considered undruggable targets and how they are being prosecuted, and how in fact undruggable targets, if you find the right approach, are in fact druggable. We're also going to look at some of the technologies and the therapeutic modalities that are used and the ways in which they are transforming oncology drug discovery. This goes beyond some of the traditional small molecule and simple antibody type approaches. We're also going to look at ways in which we can potentially minimize clinical failures, and the way in which to do that is by addressing the other part of drug discovery, which is looking at safety and toxicity, and how we can potentially address and resolve possibly unforeseen toxicities so that clinical failure is reduced. And then we're also going to look at ways in which an oncology drug's mode of action or biological context can be more thoroughly evaluated prior to taking that drug forward for clinical testing. So to start with, this is a general overview of drug discovery, and this is applicable to really any therapeutic area and not specifically just drug discovery. We simply go from identifying a target, and this may be, with respect to oncology, the association of a particular oncogene with certain tumor types. It may be a particular gene that is mutated in certain tumors or overexpressed in certain tumors. Or it may also be a component of a pathway that is activated or hyperactivated in certain tumors. We go through the process then of identifying either compounds or antibody clones, biologic drug discovery type therapeutics, typically through screening, or in the case of biologics, panning, establishing activity, and then really profiling those candidate molecules prior to taking them forward as potential candidates. Obviously, we have to test safety in GLP, and then following IND or investigational new drug approval, going then into the phases of clinical trials where we're looking initially in phase 1 at safety and tolerability, phase 2 for efficacy, phase 3 for statistical significance of efficacy, and then ultimately approval and launch, and then phase 4, which is the subsequent evaluation beyond the approval. So if we look at oncology attrition rates and where oncology drugs have failed, or looking at the better side of it, passed, we can see that oncology drugs unfortunately have tended to have a fairly high attrition rate. And attrition rates generally, as we can see here, about 60% of failures are occurring in clinical trials between phases 1 and 3. So collectively there, we're looking at the safety and toxicity side in phase 1, the tolerability of a drug, phase 2 being efficacy, and phase 3 essentially being a statistical extension of efficacy. We can unfortunately see that oncology drugs, only about 5.3% of drugs that go into clinical trials will actually end up being approved as marketed agents there. So we've got to do things that are transformative and ways in which we can address some of those unforeseen toxicities to greatly improve this rate. This rate is not currently acceptable. As I'd mentioned, if we look at causes of drug failure, this is overviewed in the pie chart on the right-hand side of this slide, and you can see a large number of drugs, approximately half of them, fail due to efficacy. So those drugs do not meet their clinical endpoint. Another large number of drugs fail in safety. So this would be the phase 1 part of clinical testing. So approximately a quarter to 1/3 of drugs fail there. And then there are other reasons that drugs either fail or are withdrawn from development. So really the two big areas are lack of efficacy and safety. So what are the ways then in which we can address these potential sources of failure in order to improve success rates and to reduce attrition when a drug goes into the clinic? If we look then at some of the current challenges with oncology drug discovery and how those tie in with those two failures, we can begin to see where new areas are being explored to help reduce those attrition rates. So from the perspective of efficacy, one of the ways forward is to identify new or novel targets that are key cancer drivers. This has long been the way in which oncology drug discovery has been conducted. A good case in point being the identification of BRAF mutations back in 2002 as being associated with a variety of cancers. And in turn, that resulted in a number of programs and subsequently approved drugs that target BRAF as a therapeutic target. Looking at undruggable targets, I never like the term undruggable. I believe that everything can be drugged. It's just a question of finding the right way to do it. One example that we will look at briefly is KRAS, long thought to be an undruggable target. But of course, as we well know, currently there are now approved drugs that can therapeutically address that target. And that obviously is a key oncogene in many cancers. Then we'll look also at the ways in which drugs can be tested on more translationally relevant in vitro and in vivo models. So what can we do there to really make that link between in vitro models and clinical efficacy? So there's a long way to go between biochemical assays and clinical relevance. But what can we do to close that gap in a translationally relevant manner? The more translational a study is, the more likely it is to result in a drug that will succeed clinically. And what can we do to improve the understanding of a drug's mechanism of action? Often that can be very important in identifying better ways forward. And so also finding the right patient. So biomarkers of response, and how can we really capitalize on that? Then other aspects of reducing attrition here are to develop drug candidates that have lower chances of unforeseen toxicities in the clinic. So looking at things like on-target versus off-target activities or toxicities, and selectivity profiling. And then profiling molecules on predictive markers of toxicities. There are ways in which we can begin to tease out or identify potential toxic liabilities. And of course, understanding and improving our understanding of drug activities in more biologically complex systems. And this also ties in with the translationally relevant aspect of the assays that we use. So if we look at various trends in therapeutic modalities, there's obviously precision medicine. The example I gave there of drugs targeting mutated BRAF are a good example of precision medicine. There is, of course, and has been for now several years, the Nobel Prize winning discovery of being able to harness the immune system and to effectively unmask the ability of tumor cells to evade the immune system. And so there are ongoing efforts in this area to really fully exploit this. And then, of course, there are new modalities. There are things like cell therapies or CAR T therapies, as they are known. Antibody drug conjugates are another way of capitalizing on the benefit of selectivity of antibodies in combination with the cytotoxicity of some of the previously used harsh cytotoxic therapeutics or even toxins, but focusing them using the selectivity of the antibody part of an ADC. And of course, small molecules do still remain a key modality despite the rise of biologics. Obviously, biologics are very important in providing a variety of different ways of targeting cancers, but small molecules are still just as important there. But there are new approaches within that, in particular combinations, and some of the new approaches with the way in which those small molecules are designed, for example, degraders or PROTACs and molecular glues. So thinking about then examples of novel targets and where we can go to look at novel targets. Historically, a number of target classes have been long associated with oncology, for example, kinases. But as we come to looking at new ways to treat cancers and addressing the question of looking at other targets, helicases are a good example. So in particular, the example here given is the Werner syndrome protein, or WRN, which is a REC-Q family helicase that is involved in the maintenance of genomic integrity. And this is shown here on the right in this schematic. So you can see that in mismatch repair proficient cells, or cells that are functioning normally, there is a normal process of DNA repair and normal proliferation of those cells. If one then impairs the WRN helicase, the DNA is still repaired because there is the mismatch repair and processes available to be able to circumvent that, and normal proliferation still occurs. However, in mismatch repair deficient tumor cells, and this is frequently seen in tumors where microsatellite instability is seen, in this particular case, the DNA is repaired and normal proliferation is seen. However, if the helicase is inhibited in those particular tumors, then the DNA becomes damaged, and this ultimately results in a cell cycle arrest and death of the tumor cells. So this represents a new way that we've seen in recent years of targeting tumor cells. So this is an example of employing a synthetic lethal rationale. There are many screens that people are currently doing to look at synthetic lethality as a way to identify new targets for drug discovery. And this, in fact, has been successful. There are now 2 small molecule drugs shown here that are presently in clinical development that target this particular helicase. So if we look at ways that we can screen and profile in this particular example for WRN helicase inhibitors, there are various ways that we can do this. We can look at the ability of a helicase through its ATPase activity. Helicases require ATP for activity. We can also look at their functional ability to unwind DNA. It's what they function to do. So we can look at ways in which DNA is unwound. We can set up in vitro assays to do this, as shown by these 2 panels here. So then when we look at a couple of different drugs, and these are those 2 drugs that are in development, we can see that they show different activities, both in ATPase activity, as well as in inhibiting unwinding, DNA unwinding processes. So this shows that we can really set up some good assays to be able to profile helicases. So much so that if you look at the panel on the right here, we've established a number of assays here that address a wide variety of different helicases, and with respect to different assays such as the ATPase activity, the unwinding activity, and also biophysical assays where we're looking at direct binding to the helicases. Now, I'd mentioned briefly earlier the concept of undruggable targets. There are many targets that have historically been thought to be undruggable. They've been thought to be central to cellular viability. KRAS is a good example of this. The real differential, of course, between normal cells, normally functioning, normally growing cells, and tumor cells is that in many cases, KRAS is mutated to a constitutively active form. So recently, there has been tremendous excitement in the field with the approval of so far 2 drugs that target KRAS, mutant KRAS. And so that really has shown that these previously thought to be undruggable targets can in fact be drugged. And the examples are sotorasib and adagrasib. And these work by covalently binding to the KRAS target within the binding pocket. So there, of course, then are ways that we can look at those different inhibitors. So we can look at things like cell-based target engagement assays. We can look, for example, also cell-based targeted protein degradation. We can, of course, look at biochemical activity assays and biochemical binding assays. So with these various kinds of assays, we can then put in place or have at our disposal a toolbox with which to profile drugs as they are developed. And of course, as I'd mentioned, one of the reasons why KRAS was previously deemed to be undruggable is because it is important and its function is important in normal cells. So there must be the ability to show selectivity for wild type versus mutant or tumor cell lines. And so if we look at the panels on the right-hand side of this slide, these show in the top left here the example of A549, which is a KRAS G12C mutant tumor cell line. And you can see the two drugs here, AMG 510, which became sotorasib, and MRTX 849, which became adagrasib. And these both show appreciable activity against this particular mutant KRAS in this cell line. If we look at A549 that has been CRISPR engineered to present the G12D mutant, you can see that these two G12C mutant-specific drugs do not affect this cell line. Whereas in contrast, MRTX 1133, which is selective specifically for the glutamate mutant here, does show activity. And then when we look at A549 cells where KRAS has been CRISPR engineered to be wild type, we see that none of the drugs show appreciable activity. So graphic demonstration that we can put in place cellular assays that not only show potency, different potency of drugs, but also the selectivity of drugs, not only for mutant types, but also the wild type. There are other approaches, of course, that we can also use, and this has become an emerging trend in drug discovery in general, but in particular in the oncology field, which is the use of structure-guided strategies. So this can, in fact, help accelerate inhibitor development because one is looking at the crystal structure of a particular target protein and then exploring potential binding pockets in which molecules or drugs can then bind, and then engineering those drugs to fit that binding pocket such that that binding would be potentially ideal. This, of course, can then be tested in those assays that we've just outlined. And again, looking at something like the G12C mutant of KRAS and the binding in this particular case of AMG 510, or what became sotorasib, versus the G12D mutant of KRAS, and in this case looking at the binding of MRTX 1133. So you can see that these different methodologies here can be applied to different mutants and thus accelerate the development of inhibitors for those mutant-specific drugs. There are other approaches that have been employed with some degree of success, and again, this is a very hot field in the oncology drug discovery area. And this is looking at bifunctional degraders, or what we know as PROTACs, and molecular glues. Now, these both ultimately result in the destruction of the target protein. So if we think actually about the ways in which often targets have been validated in a target validation sense, often RNA knockout type experiments have been conducted. Of course, if one is looking at a small molecule that is designed to inhibit, say, the catalytic activity of a potential target, a knockout doesn't necessarily give the same results as a pharmacologic inhibition. But for things like PROTACs and molecular glues that result in degradation of the target protein, this is an example where those kinds of early target validation can also potentially be very useful. So just looking at these two mechanisms then, PROTACs work by bringing it into close proximity of the protein of interest, the POI here, the oncology target of interest, using effectively a bifunctional compound. It has a target binding entity, a linker, and then an entity that binds to a an E3 ubiquitin ligase. So this brings this into close proximity with the target, results in target ubiquitination, which then ultimately targets that protein for destruction. Molecular glues work in somewhat of a similar manner, but they do this by directly binding the ligase or bringing the ligase into close proximity of the target of interest. So the ligase then can directly ubiquitinate that particular target. So again, there are various ways that we can look at these interactions from things like a biophysical approach that allow us to conduct this kind of drug discovery. So thinking about the ways in which we can screen for these kinds of inhibitors, we can look for binders that bind to a specific site. So this is an example that we've conducted looking at Wee1 kinase. So you have basically a KD that is measured for the target here to bind the Wee1 kinase. We can look at KDs for binding of a drug that binds to the E3 ubiquitin ligase. And then we can look at the two together. So we can actually do different kinds of screening to identify molecules and generate SAR then that can guide the binding of both the target, the ligase, and ultimately the two together, which results, of course, in the molecular glue idea. So this is just a quick example of where we've conducted some hit characterization for molecular glues. This is again looking at Wee1 as an example. And so you can see here we've demonstrated KD values for Wee1 alone, the ubiquitin ligase alone, and another form of ubiquitin ligase. What you'll note here is different assay methodologies that are used here: spectral shift for cereblon, or the E3 scan platform that we have for cereblon, and the kinome scan platform, which is a binding platform for Wee1. But then when we look at ternary complex formation, so this is the formation of all three as one ternary complex, we're then able to demonstrate the binding of certain drugs here to the ternary complex, but not specifically to the individual agents with any appreciable potency. So what we can see here then is we look at Wee1 as a target in a cellular context. We can actually show that these drugs do result with increasing concentration, so a dose-responsive result here in the degradation of Wee1 kinase. I'd mentioned also other modalities employing the specificity of antibodies. This is antibody-drug conjugates. So antibodies have long been used as therapeutic agents. So if we then employ their selectivity and we then conjugate them with a toxin, this allows us to then bring that toxin selectively to specific cell types, and typically human tumor cell types. So we can then create a very localized, selective, high concentration of that toxin within the tumor cells, while sparing normal cell types and systemic exposure to the drug. There are various factors that need to be considered in ADC design. It's not simply a question of linking a drug to the antibody. We have to look specifically at the antibody and how that can then bind to its target cell type. We have to look, of course, at the toxin and to make sure that the target cell type is ultimately sensitive to the toxin. But the linker provides a very important part of ADCs, and a lot of work really is focused on optimizing the linkers. Some linkers would seem to be fairly good at linking a drug to the antibody, but unfortunately they don't result in appropriate activity. They either are unstable or the drug doesn't behave ultimately the way it is desired when it gets into cells. So there are numerous ways then that one can look at the construction of an ADC, and it's not just the linker, it's looking at all aspects of the therapeutic as a whole, and really characterizing all aspects of that and optimizing it such that the ADC has the selectivity and the targeting that is needed. Now, what we're going to do now, following on from the idea of antibodies providing a certain degree of selectivity, is we're going to look at selection biomarkers that can be used to select for appropriately responsive patient populations in clinical trials. So this has been done for quite some years now. Initially, Genentech really employed this. This was quite a groundbreaking approach when they conducted trials with trastuzumab. And what they did in that particular case, trastuzumab being a HER2 targeting antibody, is they looked at breast cancer. And breast cancer, of course, being one of the cancer types where HER2 overexpression is often seen. That thus improved the chances of seeing clinical success with trastuzumab, and in fact that was seen, and it was subsequently approved in 1998 as Herceptin. But more recently, the idea has been taken even further, and Loxo Oncology, with their drug that became larotrectinib, approved in 2018, weren't so worried about the tumor type. They used a genomic predictor and basically said that any patient with an NTRK fusion would likely benefit from the treatment with their drug. And that was in fact seen to be the case. So this transformed the way in which clinical trials are done yet again by being effectively tumor type agnostic, but really letting that genetic profile guide the chances of efficacy in the clinical trial. So you can see here on the right-hand side, if you use pre-selection biomarkers, it doesn't really impact phase 1 because this is looking at toxicity, safety, and tolerability. But you can see that when using pre-selection biomarkers versus a no biomarker selection or lack of selection process, you can see that there is a dramatic increase here in the success of those drugs or the probability of success. This carries through into phase 3 trials. Obviously, having established it in phase 2, the chances of seeing it in phase 3 are increased. But of course, one is looking at a much broader population of patients in that particular case. But the effect is still seen. And so ultimately, phase 1 to approval overall is greatly improved by a little bit more than twofold. So tremendous success here in using tumor selection biomarkers to increase clinical success. So there are ways in which we can do that. If we profile a drug against a broad range of human tumor cell types, and they don't necessarily need to be cell types that contain a mutation of interest or a target of interest that is overexpressed. Really, the broader and the more diverse and agnostic that tumor cell type collection is, the better the chance of being able to demonstrate and identify selectivity for either particular tumor types, and that's shown in the top left here when looking at vemurafenib. Predictably, you can see that skin or melanoma tumor cell lines in particular are most frequently sensitive to vemurafenib, and this reflects the high presence of the V600E activating mutation within those tumors. But of course, it is seen in other tumor types, so you do see activity in other tumors. Right the way through to then, if we look at mutations, so if we screen a large number of human tumor cell lines with a particular drug, and again, this is showing vemurafenib as an example, and we segregate the cell lines and we rank order them by sensitivity, going from most sensitive to least sensitive, we can draw cutoff limits. And then within those boxes or those bins of cell lines that are sensitive or most resistant or insensitive, we can then ask statistically using genomic databases what mutations or in fact differential expression of genes correlate with sensitivity to that drug. Excuse me. So what we see here is when we look at vemurafenib, we see the association of BRAF mutation or amplification of function with sensitivity to this drug. And this is when plotting the odds ratio against the effect. So this is, of course, is what we would expect to see for vemurafenib. If we consider something like a MEK inhibitor, typically those MEK is not usually overexpressed or mutated in human cancers, not frequently at least. So, of course, then you're looking at tumor types that would be responsive to MEK inhibitors. And what we see there is the occurrence of KRAS mutations, really, because that is often driving tumorigenesis through activation of the MAP kinase pathway, which is, of course, where MEK exists. If we look at resistance, of course, in this case for vemurafenib, we see KRAS mutation here. We also see FBN2 mutation and P53 mutation. And these all make sense when you think about about a BRAF targeted therapeutic. In particular, KRAS. Of course, it's upstream of BRAF, but it can activate many more things than just the mitogen activated protein kinase pathway. It can activate survival pathways, and this is, of course, how KRAS mutant drugs can tend to get around BRAF inhibition. So examples here of where we can identify predictive genomic biomarkers, not only for sensitivity, but also resistance. That can help guide and stratify patient populations for clinical testing. And it can also potentially identify rational combinations that can be explored before a combination may be tested in the clinic. Now, if we look again at ADCs, this is really a graphic demonstration here using, again, the same type of approach, looking at a large number of human tumor cell lines with a particular ADC. And in this particular case, we're focusing on trastuzumab deruxtecan. So trastuzumab deruxtecan is an ADC that employs trastuzumab as the monoclonal antibody moiety. And when we look at its selectivity across a broad range of human tumor cell lines, you see that the vast majority of them are actually insensitive or resistant. But there is a small subset that are sensitive, and typically these are showing an overexpression of HER2, as you would expect for trastuzumab. If we look in contrast at deruxtecan is a topoisomerase inhibitor, and if we do the same kind of rank ordering of sensitivity of human tumor cell lines here, we see a typical waterfall plot that is very typical of a cytotoxic agent like deruxtecan and other cytotoxics. But again, we can say these cell lines here are what we would consider the most sensitive, and these are the most resistant. And when we look at the ADC combined, so the deruxtecan that is conjugated to trastuzumab, again, we see that it is not broadly cytotoxic across the vast majority of cell lines. So we've really then dampened down the broad spectrum non-specific or relatively non-specific cytotoxicity of deruxtecan. But we have in fact slightly expanded the population of cell lines that we see that are sensitive to trastuzumab-directed deruxtecan. Now, another factor to bear in mind here is when we look at trastuzumab's activity, its anti-proliferative activity for tumor cell lines, just showing in the bottom right here, SKBR3, which is a breast cancer cell line that overexpresses HER2, what you see then that trastuzumab shows pretty much a partial response. But and this is typical of trastuzumab is therapeutically very active because it also employs ADCC, which brings in the immune system then to destroy the tumor cells. So, of course, in this particular assay format, we don't have an immune cell component. We're looking at just at direct anti-tumor activity. But you can see when we link trastuzumab to deruxtecan in the blue, we see a much more, firstly, potent and a more complete response. And when linked with MMAE, which is a maytansinoid toxin, you see a complete response here. So this graphically demonstrates then that these cell lines that are responsive here to trastuzumab deruxtecan are showing more of a cytotoxic response with just this therapeutic alone, as opposed to the naked antibody. So a good exemplification here of the benefit of ADCs in targeting toxic agents to tumor cell lines to kill them. Now, of course, what we need to make sure, really, with any oncology drug is that it doesn't have unwanted broader consequences. So one way that we can do that is by looking at these drugs in more complex systems, more complex biological systems. And an example of this is what we call the BioMap platform. BioMap makes extensive use of normal human primary cell types, often in co-culture. So there are no tumor cells within many of these systems that we look at here. We're really focusing on the normal cell types. We have broad coverage of a variety of different tissue biology and disease states, so we can look at that broader context. And all of the biomarkers that are measured are clinically relevant translational protein biomarker endpoints. So collectively, these parameters that relate to how BioMap is run as an assay all contribute to its high physiological relevance and translatability. Now, what makes this platform very powerful is that we have tested over 4,500 agents against the platform. If we measure a range of biomarkers, typically 148 of them across 12 different models or systems, we can generate what I call a molecular phenotypic fingerprint. And that is unique for every drug. Every drug has its own profile. So having profiled over 4,500 agents, and these represent a whole variety of drugs, both approved drugs, drugs that have been into the clinic and failed for various reasons. And they can themselves yield some very valuable information. Tool compounds, mixtures, antibodies, peptides, recombinant therapeutic proteins, natural products. We have a vast range of test agents that represent a broad range of therapeutic targets and different therapeutic modalities, all of which have their own unique fingerprints. So if we test a new agent, we can then look at that database, and statistically we can do a number of things. We can identify what is the mechanism of that new agent. This gets to one of those challenges of drug discovery, is identifying how a drug works in certain cases. In recent years, there has been a lot of emphasis and re-emphasis on the use of phenotypic screening for drug discovery. In those cases, there is a very desirable endpoint. In the case of oncology, it's typically killing human tumor cells. And if you can show then a differential between killing tumor cells and not killing normal cell types, you have a promising start for a drug. But of course, with phenotypic screening, you have no idea necessarily how that drug is working. So by comparing the profile of one of those drugs with our database, we can potentially identify how that drug is working. And we have done this on numerous occasions. We can also potentially identify toxicities or potential toxicities. And a way in which we've done this is by looking at potential toxicity signatures of drugs that have similar toxicities. And so for a new test agent, we can then look at its profile and say, does it have any hallmarks that are indicative of those toxicities? We can also look at things like combinations to see whether a combination results in a safer drug, or maybe a drug that can be used at a lower concentration, or two drugs that can be used at a lower concentration, thus reducing their inherent toxicity by virtue of the fact of having lower exposure. And we can benchmark against approved drugs. So here we're actually looking in this highly translational system at how does a potential new drug look as compared with an existing therapeutic. And that may, to an extent, get to resolving failure in phase 3 clinical trials. Now, we can also look at things like ADCs, and an example here, and bear in mind that what we're looking at here are 12 different models, 12 different systems, all of which use normal human cell types. So while an ADC, in this particular case, again, trastuzumab deruxtecan, shows great activity, great anti-tumor activity, we would hope that it doesn't have great anti-proliferative or cytotoxic activity against normal cell types. So if we look then at the top here, we see trastuzumab. So trastuzumab shows a few activities here. It shows a little bit of anti-proliferative activity in this particular system. This is largely because of inhibition of vascular endothelial cell proliferation, but it is not cytotoxicity. The wide gray arrow here indicates an anti-proliferative activity. A solid black arrow would indicate cytotoxicity. And this, of course, is why certain aspects of trastuzumab therapy are seen. We also see a certain degree of anti-proliferative activity here. If we look in contrast then at trastuzumab deruxtecan, we see another couple of cases here where we see a little bit of anti-proliferative activity. But it's important to note that we don't, in any of these systems, see any cytotoxicity. And when looking at some of the other biomarkers that are modulated, in many cases, the profile is often very, very similar. We see, for example, here, and over here. This is a little bit more accentuated in this particular, with trastuzumab deruxtecan, but again, it relates to this anti-proliferative activity. But the take-home message here is then when using an ADC like trastuzumab deruxtecan, while the topoisomerase inhibitor in its own right, the deruxtecan moiety is quite cytotoxic, it isn't displaying broad-spectrum cytotoxicity here against these normal cell models. We can, of course, also use this platform to look at immuno-oncology properties, and these are models where we do, in fact, use a human tumor cell line. And when we administer a checkpoint inhibitor, what we show here is a restoration of immune response, both in a stromal model or a vascular model. So these are effectively co-culture models where we are showing the ability of things like nivolumab and pembrolizumab to restore that immune response, which is exactly how these drugs work clinically. But we can also use this platform to look at other drugs. Look at this. Erlotinib, gemcitabine, and paclitaxel. These drugs were in use long before the concept of immuno-oncology really arose, and they certainly weren't designed to be immuno-oncology agents, but they have a certain degree of immuno-oncology quality to their character. This ultimately contributes probably to their therapeutic activity and why they are very good drugs in their own right. Not only do they target their respective anti-tumor targets, but they also have an immuno-oncology component. So what else can we do to reduce attrition before clinical evaluation? We talked a lot about how failure rates are still unacceptably high. So what in vitro strategies can we use then to evaluate drugs for clinical testing and improve the success rate? There are, of course, a large number of safety pharmacology assays, and panels exist, both smaller panels for more rapid screening earlier in drug discovery, right the way through to more complex panels and more thorough panels as one gets close to candidate selection. And these remain very much a gold standard for looking at safety pharmacology. So much so that the IQ DrewSafe consortium recently, it was a consortium of pharma companies, got together and reviewed the safety targets and said, there are a few more that we need to add to some of the traditional panels. And so we have actually added those to some of the panels that we provide. So you can look at direct activity against certain targets. And examples here are some of the targets that are different between idelalisib and umbralisib, two PI3 kinase inhibitors that were both approved for use in oncology therapeutic scenarios. But you can see here, idelalisib has a little bit more of a broader profile in terms of safety targets than does idelalisib, and certainly as far as potency goes as well in this particular case. We can also look at what's called a radar plot, so we can see how different drugs compare across these panels, and we can also then use this information to assess potential organ liability. So these assays are still very, very good assays to use, but they are targeted assays, so they look at specific targets. So what can we do then to look a little bit more agnostically and broadly in perhaps something like the Biomap platform, where we're looking then at mixed co-culture models of normal human cell types? This gets to that translational ability. So as I'd mentioned, in Biomap, we've profiled a large number of reference drugs, approved drugs, many of which do have known toxicities. If we look at drugs that have the same toxicity and we compare their profiles, while each of their profiles are fundamentally different, in certain cases there are shared activities in terms of biomarkers that are modulated. So if we do this with enough drugs that have the same toxicity, and we conduct a data mining exercise, we can look statistically to identify specific biomarkers that relate to all of those drugs that have that common toxicity. And this has resulted in what we call these toxicity signatures. We've identified 9 different toxicity signatures here, and in each case we've associated them with reference drugs that are known clinically to cause those toxicities. And we also have identified pathways or biomarkers within those pathways that can contribute to those toxicities. So if we think then about identification of potential toxicity liabilities within a test agent drug, This is coming back to idelalisib. We showed that just a couple of slides ago in safety pharmacology assays, and you can see across the majority of systems here, the different models, there's very little change outside of the vehicle control, which is the grey signal at the back here. But what you see is striking activity in the BT system, which specifically looks at T-cell mediated B-cell activation. There is a degree of anti-proliferative activity here. But this is exactly what you would expect of a PI3 kinase inhibitor, PI3 kinase activity being fundamental to things like B-cell receptor activation and signaling. And that's, of course, manifested by a decrease in secretion of IgG and various cytokines associated with B-cell function. But otherwise, you see a pretty good profile here, very little activity elsewhere, a little bit of anti-proliferative activity here in this particular system, likely through some subtle modulation of fibroblast proliferation. If we then take this profile and we do that toxicity signature analysis on it, we see here just one toxicity, and this is immune suppression. And this is exactly what is seen with idelalisib clinically. And so we can then rationalize some of the subsequent clinical effects that are seen with that. In fact, idelalisib does have a boxed warning for intestinal perforation, which itself can be a subsequent result of related inflammation that results from infection, which itself comes back to this immunosuppression of B-cell activity. So consequential toxicity in this case. If we look in comparison then with umbralisib, this was an approved PI3 kinase inhibitor. Unfortunately, it was withdrawn because the long-term side effects of this outweighed the therapeutic benefit. And there was an increased risk potentially of death with this. So if we look at the umbralisib profile, we can see here the tremendous activity in the BT system, just like idelalisib, but we see a number of other activities here. And when we do the toxicity signature analysis on this, we do see immunosuppression, as we would expect, but we do also see skin irritation, organ toxicity, skin sensitization, and vascular toxicity associated with this drug. So likely contributed ultimately to those longer term effects that were seen, and ultimately its unfortunate withdrawal. If we then look at other approaches, so thinking here about some of the in vivo approaches and ways in which more comprehensive evaluation can identify the best position for a drug, an example here is sorafenib. Sorafenib was originally being developed as a RAF inhibitor, ultimately for the treatment of things like BRAF mutant-driven cancers. Unfortunately, when it was taken into clinical trials, it didn't fare so well, didn't really show its desired efficacy endpoints. When you look at its activity in a panel of normal human tumor cell lines for anti-proliferative activity, you can see here it has activity against a certain number of cell lines, but when you color those cell lines in red here, as whether they contain BRAF activating mutations or c-KIT overexpression, which can sometimes also drive signaling through the BRAF pathway, there really isn't a strong statistical correlation with activity. In contrast, when we look at vemurafenib, we do see that association. You see strong activity in the large majority of BRAF mutant cell lines and even some of the c-KIT overexpressing cell lines, and also NRAS mutant cell lines, which can also drive signaling through BRAF. These show some degree of activity here. So if we then look at sorafenib, ultimately sorafenib did show great utility and is now approved for use in things like renal cell carcinoma and hepatocellular carcinoma. If we look here at this particular model, this is an in vivo tumor xenograft model, where we're looking at sorafenib here in blue, and this is in fact using a mouse rancor model. So this is a syngeneic model in mice. It shows great activity. In contrast, anti-CTLA4, anti-PD-L1, and anti-PD-1, so these are biosimilars for checkpoint inhibitors, show some degree of activity, but sorafenib is superior to them here. And of course, this reflects exactly where sorafenib found its therapeutic utility. So really being thorough and comprehensive in the evaluation of drugs going forward can really identify the best tumor types for them. So just in summary then, we've seen how the various challenges of oncology drug discovery can present significant hurdles to identifying efficacious and safe drugs. But I always like to think that those hurdles are not insurmountable. And really, by use of a broad palette of different assays, technological approaches, and strategic approaches, we can begin to identify drugs to address those challenges within drug discovery. And at Eurofins, we do offer a broad palette of different assays ranging from chemistry and biotherapeutics right the way through in vitro pharmacology, translational cell assays. We looked at some of those in this webinar. In vivo safety and efficacy models, in vitro safety, and again, just to reiterate, those models are still perfectly good models, and they are very good at looking at safety targets that we know, but in combination with translational assays looking more agnostically, we can identify potential safety and toxicity risks that we wouldn't expect. And of course, ADME. So collectively, looking at all of these approaches is important in identifying efficacious and safe drugs. We can, in fact, offer these as an integrated drug discovery program. So we have ultimately a broad collection of assays here, a diverse portfolio that really at any level can help address challenges within oncology drug discovery. We've talked about some of the novel target types here and different assay methodologies, ADCs and things like this. We really are very open and available to working with anybody on their programs, and we love a challenge. So we love to be able to help programs by employing the technologies that we have. And in fact, many of these technologies have been developed as a result of challenges in oncology drug discovery. And of course, as you all, as oncology drug discoverers, seek new drugs, and there are new challenges that we encounter, we continuously expand our capabilities to meet those challenges. So we work with you on this challenge for the greater good of humankind. If you'd like to learn a little bit more about this, please feel free to reach out. We have QR codes here. We have a number of promotional material. And of course, I would be remiss if I didn't thank you all for your attention. 감사합니다. And I would also like to thank not only the BRIC organizers for the opportunity to give this webinar, but our distributor in Korea, Chaeyoung Laboratories. So thank you all very much. I'd be happy to answer any questions.